All right, good morning. Are we? Good morning. Yes, Vivek Singh, you're the first one. Good morning, Nivedita. Vandana Singh, good morning. Good morning. Yes, and ready for another long session today? Yes, tell me about the audio and the video. Are you all okay with it? Are you all okay with it? Yeah, good to see you too, Ashwat. Yes, though I can only read your messages, but still very, very okay. Good evening, Akash. Good morning, bhai. Yes. Okay. All right. So tell me about the audio video. Is everything fine? Are you able to see well as hear me properly? Okay, great. So can we start? And I think let's not waste any time and let's start. Though I'm going to take a couple of doubts. I've been getting a lot of your messages and emails on. There were some common doubts, and let me just tell you about them. Right? Yes. So let's just see this. Now, as far as the first doubt, which was a common doubt, which I got a lot of questions on, is the relationship between mole and osmole. So I'm just taking the doubts first. Yes, doubts from the last class. Number one was, what is the relationship between the moles and mole and osmole? So please remember that one osmole of any substance is equal to 1 mole of that substance divided by the number of freely moving particles liberated in solution, right? Number of freely moving particles liberated in solution, right? Now, for example, when I took the example of sodium chloride. Sodium chloride, when you dissolve it in water, splits into 2 particles. Or let's take the example of potassium chloride. Potassium chloride, when you dissolve it in water, splits into 2 particles. Calcium chloride, on the other hand, will split into 3 particles, right? So let's see this. Now, 1 osmole of, let's say, sodium chloride is equal to 1 mole of sodium chloride divided by the number of freely moving particles liberated in solution, and that was 2, right? So, in other words, 1 osmole of sodium chloride is equal to 0.5 moles of sodium chloride, or in other words, 0.5 moles of sodium chloride is equal to 1 osmole of sodium chloride, right? So in other words, 1 mole of sodium chloride will be equal to 2 osmoles of sodium chloride. Yes. So this was a common doubt which I had been asked, that how is it possible? Yes. One way is, so the question is, 1 mole of sodium chloride is equal to how many osmoles? So what do? Either you could have cross-multiplied this over here, yes, or follow what I've done. I've just sort of tried to explain it to you, why 1 mole of sodium chloride is equal to 2 osmoles. So remember, mole of any substance will be equal to the number of freely moving particles. If it liberates 2 freely moving particles, 1 mole of that substance will be equal to 2 osmoles of that substance. If it liberates 3 freely moving particles, 1 mole of that substance will be equal to 3 osmoles of that substance. So this is just a doubt which was a common doubt from last class. Okay. Now, another doubt which I wanted to discuss with you, and one thing, please add it to your notes. Please add it to your notes, and that is how to calculate the plasma osmolality. Now, this was something which I forgot to tell you. How much is the plasma osmolality in milliosmoles per liter? How much is it going to be? This is 2 into sodium plus potassium. Sodium and potassium may be in millimoles per liter or in milliequivalents per liter. How to calculate the plasma osmolality? Right? How to calculate the plasma osmolality? This is 2 into sodium plus potassium, that is millimoles per liter or milliequivalent. Please add this to your notes. Please add the following. Please add this to your notes. How to calculate the plasma osmolality, that is 2 into sodium plus potassium plus 0.055 into glucose. Glucose is in milligrams per deciliter, right? Milligrams per deciliter. 0.055 into glucose plus 0.36 into blood urea nitrogen, which is also in milligrams per deciliter, right? Milligrams per deciliter. So see this once again. How do you calculate the plasma osmolality? Plasma osmolality is 2 into sodium plus potassium plus 0.055 into glucose plus 0.36 into blood urea nitrogen, right? So this is millimoles per liter or milliequivalents per liter. And like I said, glucose is in milligrams per deciliter. Why do I multiply it by 0.055? To convert the milligrams per deciliter into milliosmoles per liter. Right? And similarly, blood urea nitrogen, which is given in milligrams per deciliter, to convert it into milliosmoles per liter, you have to multiply it by a correction factor of 0.36. Right? Now, based on this, you had a neat question, right? You had a neat question. There was a neat question which was asked. Dhara Shakti, I am Maz Khan, Barkha Lodiwal, I'm just some small topic which I forgot to mention last time. I'm doing it now, so don't add it to your notes. That's all that I've asked you, right? Okay. Now, there was a neat question which was asked, and that said that sodium is 140 millimoles per liter, potassium is 5 millimoles per liter, glucose is 5 milliosmoles per liter. Blood urea nitrogen is 5 milliosmoles per liter. How much is the plasma osmolality? How much is the plasma osmolality? Right? Sodium is 140 millimoles per liter. Potassium is 5 millimoles per liter. Glucose is 5 milliosmoles. Blood urea. How much is the plasma osmolality? Yes. Now, Ajay Kumar Reddy, what about chloride ion? You must understand, we have multiplied sodium and potassium into 2. Why did we multiply into 2? Because along with sodium and potassium, there are anions which are associated, including chloride and bicarbonate. So chloride and bicarbonate gets included in this factor of 2. Have you understood? Yes. Now, this is, now plasma osmolality, now you will take 2 into sodium plus potassium. Right? Now, in this case, there is no need to multiply by that correction factor of 0.055 because already glucose is given in 5 milliosmoles. So there is no need to multiply by the correction factor. So it will be plus 5 into plus 5 plus 5. So this is 2 into 145 plus 10, 290 plus 10, which is 300 milliosmoles per liter. Right? So please see this. This is an important point which I forgot to mention last time, how to calculate the plasma osmolality. Again, Kanishk, we have already covered this. It should be per kg or per liter simply because this is per liter, per kg can be used interchangeably because we are talking about plasma where the solvent is water. Have you understood it? We've done that already, Kanishk. You have to look up your old notes. Now, this is what I wanted you to add, how to calculate the plasma osmolality. 2 into sodium plus potassium by 2. Why do we multiply sodium and potassium by 2? Because, like I said, sodium and potassium are the cations which are associated with anions. We multiply glucose by 0.055 because glucose in milligrams per deciliter, it has to be converted into milliosmoles per liter. Blood urea nitrogen by a correction factor of 0.36 to convert it from milligrams per deciliter into milliosmoles per liter. And this was a neat question which was asked. And this is PUN is blood urea nitrogen, Shri Harsha Kavuri. This is blood urea nitrogen. Yes? All right. So this was another point which I wanted you to know. Now, the next important point. Now, Chinmay Prakash Trivedi, you've asked a very basic question. Maximum conductance Nernst potential. You are asking me, are they the same? How can they be the same? Conductance is a different term. Potential is measured in millivolts. Conductance is the property of the membrane. That means when I say membrane conductance, that means how many ions are passing through that membrane at a particular time. Whereas membrane potential or the Nernst potential is measured in terms of millivolts. So there are two different things. Have you understood? All right. Now the next important point here is one more important doubt which I want to take up from all right. Now, so this is as far as your from your last notes are concerned. Okay. Now, the next thing, let's continue from where we left last time. Okay. Yuvika, Purana's video nahi dekha? Sign wala problem nahi sort out hua aapka? Okay. All right. So let's go on to our next topic where we left last time, and that is factors affecting the velocity of conduction of a nerve impulse. Right? Factors affecting. Factors affecting velocity of conduction of a nerve impulse. Factors affecting velocity of conduction of a nerve impulse. This is where we left last time, so let's continue from there. The first factor which affects the velocity of conduction of a nerve impulse is the nerve diameter. The nerve diameter. Right? Now, how does it affect the velocity of conduction of the nerve impulse? Let's see this. Now, more the diameter, more the surface area, more will be the total number of sodium channels. Total number of sodium channels. And faster will be the velocity. Larger the diameter, larger will be the total surface area. Therefore, the total number of sodium channels will now be higher, and therefore, the faster the velocity. So, the first factor which affects the velocity of conduction is the diameter of the nerve fiber. Right? Diameter of nerve fiber. More the diameter, more the surface area. More the diameter, more the surface area, more will be the total number of sodium channels, therefore faster will be the velocity, right? Now, for example, you know that there are 3 types of nerve fibers, that is type A, type B, and type C. This is type A, this is type B, and type C. So the fastest velocity will be in the type A nerve fiber. Type A nerve fiber will have the maximum velocity. This will have the fastest velocity simply because, like I said, the larger the diameter, more the surface area, more will be the total number of sodium channels, and faster the velocity. Now, there is another way to understand. This was a simple way to understand the effect of diameter on velocity. Another way to understand is when I look at a large diameter nerve fiber, consider that this is made of smaller diameter nerve fibers in parallel to each other. Yes, a large diameter nerve fibers, consider that the large diameter nerve fiber, consider that a large diameter nerve fiber is made of smaller diameter nerve fibers in parallel with each other. Smaller diameter nerve fibers in parallel with each other. Right? Smaller diameter nerve fibers in parallel with each other. And whenever you have anything in parallel with each other, what happens with the total resistance? Total resistance is 1 upon R is equal to 1 upon R1 plus 1 upon R2 plus 1 upon R3 and so on and so forth. So basically what happens with the total resistance? The total resistance will decrease. If you remember your physics, whenever you have circuits in parallel, the total resistance decreases. When you have something in series, the total resistance increases. Right? Here, the total resistance now decreases. So when you look at a large diameter nerve fiber, consider that it is made of smaller diameter nerve fibers in parallel with each other. So now that means there is a decrease in what is known as an axoplasmic resistance. There is a decrease in the total resistance, which is known as the axoplasmic resistance. And axoplasmic resistance can be written as R small a, axoplasmic resistance, or it can also be written as R in, resistance inside a nerve fiber. So the first factor, so what am I discussing over here is, first factor is affecting the velocity of conduction, like I said, is diameter. Diameter of the nerve fiber is the first factor which controls the velocity. More the diameter, more the surface area, more the total number of sodium channels, faster the velocity. And I discussed with you that there are 3 types of nerve fibers, type A, type B, type C. Type A has got the largest diameter, maximum surface area, therefore the maximum surface area, maximum number of total number of sodium channels, fastest velocity. Another way to understand what is the effect of diameter on velocity is, consider that a large diameter nerve fiber is made of smaller diameter nerve fibers in parallel with each other. And that means, and whenever you have anything in parallel, I've just told you that the total resistance decreases. So a large diameter nerve fiber has a low axoplasmic resistance. It has a low axoplasmic resistance, and axoplasmic resistance can be written as RA or R in. And if the resistance is less, what will happen to the velocity? Faster the velocity. Velocity less the resistance, faster will be the velocity. Right? So I've explained the effect of diameter in 2 ways. One was a simple way. I told you that more the diameter, more the surface area, more the total number of sodium channels. And this is the second one, where I've said that the larger diameter nerve fiber will have a lower axoplasmic resistance and faster velocity. Right? So that means there is a question here which I'm going to ask you, and that is, a large diameter nerve fiber has a low RA or a high RA. Low RA, RA is axoplasmic resistance, or a high RA. So the answer will be low RA, and therefore a faster velocity. Right? A larger diameter nerve fiber has a low RA and therefore a faster velocity. Right? So 2 ways to understand the effect of diameter. Larger diameter nerve fibers will have a faster velocity of conduction. Right? Smaller diameter nerve fibers will have a slower velocity of conduction. Second factor which affects the velocity of conduction is the presence of myelin. Presence of myelin. Now, myelin is produced by Schwann cells in the peripheral nervous system. This is produced by the Schwann cells in the peripheral nervous system and the oligodendrocytes in the central nervous system. Right? In the peripheral nervous system and the oligodendrocytes in the central nervous system. Right? Now, how is the oligodendrocytes in the central nervous system? Yes. Now, how is myelin produced? Basically, if you see, if this is a nerve fiber, yes, if this is a nerve fiber here, the Schwann cell will wrap itself around like this. It goes round and round the nerve fiber, right? It wraps itself around the nerve fiber, right? It goes round and round the nerve fiber. In fact, it goes almost 100 times. The Schwann cell goes around the nerve fiber almost 100 times. And these successive layers, these layers of the cell membrane of the Schwann cell, they become myelin, right? Myelin, Schwann cell, the membrane of the Schwann cell, is rich in lipids, sphingomyelin, right? And so this is a lipid, the myelin will be a lipid-rich structure. This is a lipid-rich structure, right? This is lipid. Myelin, how is myelin produced? The Schwann cell in the peripheral nervous system, the Schwann cell wraps itself around the axon. It goes around the axon at least about 100 times. And these successive layers of Successive layers of the the cell membrane of the Schwartz cell, they get compacted, and that forms the myelin. Myelin is a lipid-rich structure, right? Lipid-rich structure. Myelin acts as an insulator. It acts as an insulator, right? Myelin acts as an insulator, and when it acts as an insulator, it increases the membrane resistance. It increases the membrane resistance. Membrane resistance is written as Rm. Rm. It increases the membrane resistance. Myelin, it acts as an insulator, and that in turn increases the membrane resistance, right, which is known as Rm, right? It increases the membrane resistance. So in other words, if I ask you a question, right? Like I said, see, why does myelin, which like acts as an insulator, increases the membrane resistance? It is like copper wires. Copper wires have a coating of plastic, isn't it? So what is the role of that plastic? The plastic coating that increases the resistance. So it decreases flow of current, isn't it? Normally, when you look at a wire, any wire for that matter, will have a coating of plastic. And the plastic increases the resistance and decreases the flow of current. So myelin is doing something like that. It is increasing the membrane resistance. But if it increases the membrane resistance, that means the velocity of conduction should reduce. But that does not happen, right? If you look at a myelinated nerve fiber, right? If you look at a myelinated nerve fiber, unlike a wire, has got, now if I look at a myelinated neuron like this, if I measure the total membrane resistance, it has a high membrane resistance. Yes, it has a high membrane resistance, which is known as Rm. If I measure the membrane resistance, it is going to be high membrane resistance. But you know that a myelinated nerve fiber has the presence of nodes of Ranvier, isn't it? Nodes of Ranvier are areas where there is no myelin. Between two nodes of Ranvier, the myelin is produced by one Schwann cell, right? So what are these nodes? Nodes are areas where there is no myelin. So what will happen to the nodes? Nodes are areas of low resistance. Total membrane resistance of a nerve fiber will be myelinated nerve fiber will be very high. But nodes are areas of no myelin. So that means these are areas of low resistance. And everything takes the path of least resistance, isn't it? Everything takes the path of least resistance. We do the same thing. We always take the path where the resistance is less. And that's exactly what the action potential does. Action potential, which is generated at the initial segment, now jumps from one node to the other, from one area of low resistance to another area of low resistance. Can you switch off the fan? It jumps from one area of low resistance to another area of low resistance, and this is known as saltatory conduction. This is known as saltatory conduction. It jumps from node to node, and that is how it has a higher velocity, right? So even though a large diameter nerve fiber has a high membrane resistance, but in spite of the high membrane resistance, it has a higher velocity of conduction. Why a higher velocity of conduction? Because, like I said, the action potential will jump from one area of low resistance to another area of low resistance. Which are the areas of low resistance in a myelinated nerve fiber? The nodes of Ranvier. Remember, nodes of Ranvier are just 1 millimeter apart. 1 millimeter apart. The action potential is jumping from one node to the other, which is known as saltatory conduction. Right? So these are the two factors which are affecting the velocity of conduction. Number one, I told you diameter, more the diameter, more the surface area, more the total number of sodium channels, faster velocity. Another way to understand the effect of diameter was, I told you a large diameter nerve fiber has a low axoplasmic resistance. Low axoplasmic resistance can be written as RA or Rin. This is not Q, this is A, small a, right? Small a, RA or Rin, so faster velocity. We just discussed that the large diameter nerve fiber has a low axoplasmic resistance, therefore a faster velocity. Myelin, second factor which affects the velocity of conduction is myelin, presence of myelin. Myelin is produced by Schwann cells in the peripheral nervous system, oligodendrocytes in the central nervous system. Myelin is produced by the Schwann cell, which wraps itself around the axon. So these are successive layers of cell membrane of the Schwann cell. They get compacted, and that forms the myelin. Myelin is a lipid-rich structure. It acts like an insulator. And if it acts like an insulator, it increases the membrane conductance. So large diameter nerve fibers have a high membrane resistance, but in spite of that, a higher velocity of conduction. Why? Because of saltatory conduction. So let's have a look at this question. Now let's see this. A large diameter nerve fiber. A large diameter nerve fiber has axoplasmic, large diameter myelinated nerve fiber has large diameter myelinated nerve fiber has dash RA dash RM dash RA dash RM. So this becomes low RA, I've already discussed with you, low axoplasmic resistance, but a high membrane resistance. Total membrane resistance of a myelinated nerve fiber will be high. But I've told you, in spite of a high membrane resistance, it has a higher velocity of conduction because of saltatory conduction. Right? Its action potential is jumping. Now, a very common doubt which arises over here is, if I look at an unmyelinated nerve fiber, an unmyelinated nerve fiber has a low unmyelinated nerve fiber has a low membrane resistance. It has a low RM, isn't it? It does not have myelin, therefore it has a low RM. But and if it has a low RM, it should have had a faster velocity of conduction. Lesser the resistance, faster the velocity, right? But in fact, we know that unmyelinated nerve fibers have got a low velocity of conduction. And that is also not very difficult to understand, because now what happens is, now the action potential which is generated here, the action potential now has to travel along the entire length of the axon, which is going to take much more time. So an unmyelinated nerve fiber has a low RM, and in spite of that, a lower velocity of conduction, even though it sounds paradoxical. Have you understood? We should be able to appreciate this. A myelinated nerve fiber will have a faster velocity because of saltatory conduction, because the action potential is jumping from node to node, from one area of low resistance to another area of low resistance. An unmyelinated nerve fiber has a low membrane resistance, but in spite of that, a lower velocity. Sounds paradoxical, but we know the reason, because now the action potential has to cover the entire length of the axon, which is going to take much more time. Right? So this is your unmyelinated nerve fibers will have a lower velocity of conduction. Right? Let's have a look at another question. Yes? Let's have a look at another question. And that is, fastest velocity will be present in a nerve fiber with. Fastest velocity will be present in a nerve fiber with a high Rm by Ra ratio or a low Rm by Ra ratio. A high Rm by Ra ratio or a low Rm by Ra ratio. Right? Fastest velocity will be seen in a nerve fiber with. Now, this will be a high Rm by Ra ratio. High Rm by Ra ratio, how do you get that? How do you get a high ratio? If the numerator is high and the denominator is low. When is the numerator going to be high? If it is a myelinated nerve fiber. When is the new denominator or the Ra going to be low? If it is a large diameter nerve fiber, and we've already discussed that the large diameter myelinated nerve fibers will have a faster velocity of conduction when Rm by Ra ratio is high, right? Rm by Ra ratio is high, right? Okay. Now, let's look at a third electrical property, which is called the membrane conductance. Membrane conductance. Membrane capacitance, not conductance, capacitance. Right? We've discussed the effect of diameter. We've discussed the effect of myelin. Yes, myelin on the membrane resistance. Okay. Now, let's have a look at a third electrical property which affects the velocity of conduction, and that is the membrane capacitance. Now, what is capacitance? Now, capacitance, when you did in physics of class 11 and 12, capacitance was the ability to store charge. Yes? You remember studying about capacitors, conductors, capacitors, right? Now, you must understand a nerve fiber is not like a copper wire. It is not a wire. Have you understood? In fact, nerve fibers by themselves, they are poor conductors of electricity, right? They are poor conductors of electricity, right? Poor conductors of electricity. They're not meant to conduct electricity. Please understand, it's a poor conductor. But if I apply this concept of capacitance to a nerve fiber, now please remember that the lipid bilayer of a cell membrane, lipid bilayer of cell membranes, this acts as a parallel plate capacitor. It acts like a parallel plate capacitor, right? The lipid bilayer of membranes, it acts as, please understand, it is not a typical capacitor which you did in physics. Have you understood? It simply acts as a parallel plate capacitor. Right? It acts as a parallel plate capacitor. In other words, cell membranes will have a high membrane capacitance. They have a comparatively high membrane capacitance. But if I add myelin, cell membranes by themselves have a high membrane capacitance because, like I said, the lipid bilayer of cell membranes, it acts as parallel plate capacitor. So cell membranes by themselves have a high membrane capacitance. But the moment you add myelin, you increase the membrane resistance. There is an increase in the membrane resistance, which I have just discussed it with you, which was known as Rm, but you decrease the membrane capacitance. Right? Please understand this. When you add myelin, you are increasing the resistance, but you are decreasing the membrane capacitance. Yes, you are decreasing the membrane capacitance, right? If you remember this, then it becomes very easy for us to answer questions, and that is a large diameter myelinated nerve fiber has. A large diameter myelinated nerve fiber has dash RA. What was RA? Axoplasmic resistance. Dash RA, which is axoplasmic resistance. Dash RM, membrane resistance. Dash capacitance. Low, high. So starting with axoplasmic resistance, it is large diameter, therefore low axoplasmic resistance. It is a myelinated nerve fiber, therefore high membrane resistance. It is a myelinated nerve fiber, therefore low membrane capacitance. A large diameter myelinated nerve fiber has a low axoplasmic resistance, high membrane resistance, low capacitance. Have you understood? Please remember, nerve fibers are poor conductors of electricity. Somebody's asking me why. Is a nerve fiber meant to conduct electricity? No. It is not like a copper wire. By itself, it's a poor conductor of electricity. It is not meant for conducting electricity. It is meant for conduction of action potentials, biological phenomena. Have you understood? So when you use this, Sakshi Patidar, I cannot use directly inversely proportional relationships here. It is not as simple as that. Have you understood? You cannot make oversimplified things like this. You have to understand the concept. Large diameter nerve fibers, low axoplasmic resistance, myelinated nerve fibers, high membrane resistance, myelinated nerve fibers, low capacitance, right? Then it is, there was another question which was asked that the fastest velocity is seen in nerve fibers with. Fastest velocity is seen in nerve fibers with dash resistance, dash capacitance, right? Dash resistance, dash capacitance, right? Dash resistance means, now if it just says resistance, it hasn't specified is this axoplasmic resistance or is this membrane resistance. If it has not specified which type of a resistance is it, we have to take it for granted this is the membrane resistance, right? Now that means fastest velocity will be seen in a nerve fiber with high resistance and low capacitance. Please remember, myelin will increase the membrane resistance but reduce the capacitance. A cell membrane by itself has a high capacitance, but the moment you add myelin, you are increasing the resistance and decreasing the capacitance. And myelinated nerve fibers have faster velocity. So high resistance and low capacitance. You cannot have low resistance, low capacitance, or high resistance, high capacitance. It's not myelin. The presence of myelin will increase the membrane resistance and decrease the membrane capacitance. By default, if it says only resistance, by default, you have to take into consideration the membrane resistance. Right? So let's see this once again. What is the effect of capacitance? Please remember, the lipid bilayer of cell membranes acts as a paddle plate capacitor. The moment you add myelin, so cell membranes by themselves have a high membrane capacitance. The moment you add myelin, you will increase the resistance but reduce the capacitance. So I did this question with you that a large diameter nerve fiber has a low axoplasmic resistance, high membrane resistance, and therefore a low capacitance. Overall capacitance will be low, right? Fastest velocity, therefore, will be seen in the nerve fiber with high resistance and low capacitance. Now, what is the clinical importance? Clinical importance of knowing this. Why do I want to know all this? Remember, resistance and capacitance for nerve fibers can be measured. Yes? So that means if there is a demyelinating disorder, right? Any sort of a demyelinating disease, demyelinating disorder, what will happen to the resistance? And what will happen to the capacitance of the nerve fibers? The resistance, demyelinating disorder. Now there's loss of myelin. So membrane resistance will now reduce. Yes. But what will happen to the membrane capacitance now? Lack of myelin, membrane capacitance will now increase. So there will be a reduction in the resistance and increase in capacitance. Have you understood? The best way to remember this is, please understand that the membranes, the cell membrane, the lipid bilayer is acting as parallel plate capacitors. It is not a typical capacitor, what you have learned in physics, but it is acting like a parallel plate capacitor. And we can measure capacitance of a nerve fiber. So cell membranes by themselves have got a high capacitance, but the moment you add myelin, you are increasing the resistance and lowering the capacitance, overall capacitance. So when I look at a large diameter myelinated nerve fiber, a low axoplasmic resistance, high membrane resistance, low capacitance. Membrane resistance increases, capacitance reduces. Fastest velocity will be seen in a nerve fiber with high resistance. If it says only resistance, we have to take RM into consideration by default. What is the clinical importance? What will happen in demyelinating disorders when there is loss of myelin, for example, multiple sclerosis? Yes, this is in MS patients, the resistance will reduce, capacitance will increase. Okay? All right. Okay, no, Rakshanda, we do not use compliance here, we use capacitance. This is capacitance, similar to that concept of physics which you have done, but not absolutely like what you dealt with in physics, but definitely we are applying that concept here. So this is your membrane capacitance. Okay? Sagita J, I said unmyelinated nerve fibers, no myelin, less membrane resistance, but less velocity. Why? Because the action potential has to cover the entire length of the axon. If it has to cover the entire length of the axon, it will take more time, so velocity reduces. Okay? All right. Now, this is as far as your 5 factors affecting the velocity of conduction. We discussed what are the factors affecting velocity of conduction. Diameter. Large diameter nerve fibers have low axoplasmic resistance, therefore faster velocity. We discussed the effect of myelin, and I told you the best way to remember the effect of myelin is that myelin will increase the membrane resistance but reduce the membrane capacitance. Right? It increases membrane resistance but reduces the membrane capacitance. All right. Now, the next is, let's have a look at the classification of nerve fibers. Let's see classification of nerve fibers. Classification of nerve fibers. Now, as far as classification of nerve fibers, there are 2 types of classification of nerve fibers, and that is, number 1, you have what is known as the Erlanger and Gasser classification. Erlanger and Gasser classification. Right? There are 2 classifications of nerve fibers which we have to do, and that is Erlanger and Gasser classification, and you have what is known as the Lloyd and Hunt's. Lloyd and Hunt's numerical classification. Lloyd and Hunt's numerical classification. There are 2 types of classification of nerve fibers, Erlanger and Gasser classification and Lloyd and Hunt's numerical classification. Right? Numerical classification. Erlanger and Gasser classification, now this is meant for all types of nerve fibers. It is meant for sensory, it is meant for motor, it is meant for autonomic. Right? Sensory, motor, autonomic. It is meant for all type of nerve fibers. Lloyd and Hunt's numerical classification, this is only meant for sensory. Only meant for sensory. Only for the sensory nerve fibers. Only for the sensory nerve fibers. Okay. Erlanger and Gasser classification is for sensory, all types of nerve fibers, whether they are sensory, motor, or autonomic. And Lloyd and Hunt's numerical classification will be only for sensory. Now, in Erlanger and Gasser classification, the nerve fibers are divided into type A, type B, and type C. Type A, type B, and type C. And in sensory in Lloyd enhanced numerical classification, the nerve fibers are divided into type 1, type 2, type 3, type 4. Right? Now, if I look at, remember, Lloyd numerical classification is only for sensory neurons. It is not for the motor fibers. You cannot use number for motor fibers. Numerical classification is only meant for sensory. Now, remember, A and B fibers are myelinated. C is unmyelinated. C is unmyelinated. So that means an A is the largest diameter. So as you go from A to C, the diameter reduces. A is the largest diameter, C is the smallest diameter, right? A is the largest diameter and C is the smallest diameter. A and B are myelinated nerve fibers and C is a unmyelinated nerve fiber, right? Now, so as you go from A to C, there is going to be not only a decrease in diameter, there is also going to be a decrease in the velocity of conduction. Fastest conducting nerve fibers are type A because type A is large diameter and myelinated. Slowest conducting will be type C because type C is smallest diameter and unmyelinated. We've already discussed in myelinated nerve fibers, faster velocity of conduction because of saltatory conduction. Yes, unmyelinated nerve fibers will have a slower velocity of conduction because what happens is the action potential, like I told you, I drew a diagram and I told you it has to cover the entire length of the axon. Right? Now, this type A fiber is further subdivided into A alpha, A beta, A gamma, and A delta. A alpha, A beta, A gamma, A delta. So let's have a look at a little more detail. Now, let us see A alpha fibers first. Now, A alpha fibers can be motor, they can be sensory, right? A alpha fibers, they can be somatic motor. We've already discussed this briefly last time. Remember I told you the spinal motor neuron is also known as the A alpha motor neuron. It is also known as the anterior horn cell spinal motor neuron. So that is your somatic motor. Now remember, this nerve fiber, which is a motor nerve fiber, does not have a number. It is not present in, does not exist in the numerical classification. Okay? Anjali Mahalala, small diameter and myelinated nerve fiber hai hi nahi. There is no fiber which is small diameter and myelinated. So your question becomes theoretical when there are no nerve fibers which are small diameter and myelinated. Isn't it? I've just explained it to you. Large diameter are myelinated. Small diameter is unmyelinated. Okay? Okay. So somatic motor. A alpha is somatic motor. Somatic motor. And A alpha can also be sensory. There will be some fibers which are sensory. So the sensory is for proprioception. What is proprioception? Proprioception is position sense. Proprioception. And the fibers which are meant for proprioception, so proprioception is a sensation, these will be known as type 1 fibers in the numerical classification, right? Numerical classification, right? So A alpha fibers can be motor, they can be sensory, and sensory, they are meant for proprioception. And this was a question which was asked, which is the fastest conducted sensation? Fastest conducted sensation is going to be proprioception. Yes, this is the fastest conducted sensation. Now, the type 1 fiber is further subdivided into type 1A and type 1B. Type 1A and type 1B, right? Type 1A and type 1B. Type 1A is from the muscle spindle. Type 1A is from the muscle spindle. There are two important receptors for proprioception, that is muscle spindle and the Golgi tendon organ. So type 1B fiber is from the Golgi tendon organ. This is the sensory fiber from the Golgi tendon organ. 1A is from the muscle spindle and 1B is from the Golgi tendon organ. So A alpha fibers can be motor fibers. Motor fibers will not have any number they do not have any number in the numerical classification. A alpha fibers can be for proprioception. Proprioception fibers, A alpha fibers, which are for proprioception, will have a corresponding number. The corresponding number in the numerical classification is type 1. Type 1 is further subdivided into 1A and 1B. 1A is the sensory fiber from the muscle spindle. 1B is from the Golgi tendon organ. A beta fiber. A beta fiber is purely sensory, and A beta fiber is meant for fine touch. This is meant for fine touch. This is meant for fine touch. This is meant for vibration. This is meant for vibration. This is also meant for deep pressure. This is meant for deep pressure. Deep pressure such as poking, right? Deep pressure such as poking. And this is also meant for proprioception. This is meant for proprioception. But proprioception only from the muscle spindle, not from the Golgi tendon organ. And this is the corresponding number, the numerical classification is type 2. A beta is meant for fine touch, it is meant for vibration, it is meant for deep pressure such as poking. It is not meant for pressure on the skin, but for deep pressure such as poking. And proprioception only from the muscle spindle. This is the type 2 fiber. Then you have A gamma. A gamma is motor to the muscle spindle. This is the motor fiber to the muscle spindle. This will have no number in the numerical classification because I said that no number in the numerical classification. Why so? This is because motor fibers do not have any numbers. It's only the sensory. A delta fibers. A delta fibers are meant for fast pain. There are two types of pain. There is a fast pain and there is a slow pain. This is meant for fast pain, as also for temperature. In temperature, it is meant only for cold sensation, and this is also meant for some mechanoreception. Some mechanoreception. Some mechanoreception, right? And this becomes my type. Mechanical sensation means different forms of touch, right? Mechanical sensation, some mechanoreception. So A delta will be type 3 fiber. B. Type B fiber is preganglionic autonomic. Preganglionic autonomic, right? These are preganglionic autonomic. When they have any numbers in the numerical classification? No. Right? Preganglionic autonomic, right? C. C fibers can be autonomic, so they are postganglionic sympathetic. Postganglionic sympathetic, and they can also be sensory, right? They can also be sensory. Sensory would mean they are meant for crude touch. Crude touch. Right? This is meant for itching sensation. It is meant for slow pain. We will be discussing fast pain and slow pain in CNS, right? So don't worry, it's just the introduction right now. Slow pain. This is meant for temperature. In temperature, it is meant for cold and warmth. And this is also meant for pressure. Please remember the difference between deep pressure and pressure. Deep pressure is A beta, pressure is C. So this becomes my type 4 fiber, right? Type 4 fiber. Don't worry, fast pain and slow pain will be done in CNS. So don't worry about it right now. For the time being, please remember fast pain is carried by A delta, slow pain is carried by C, right? So this is as far as your different, what are the sensations, what are the functions of different types of nerve fibers. Let's go through them once again. A alpha, like I said, can be a motor fiber or it can also be sensory. Motor fiber will not have any number in the numerical classification. Sensory, it is meant for proprioception. Proprioception is the fastest conducted sensation. For the fiber which is carrying proprioception is type 1, and type 1 is further subdivided into 1A and 1B. 1A is the sensory fiber from the muscle spindle, 1B is the sensory fiber from the Golgi tendon organ. A beta is meant for fine touch, for vibration, for deep pressure such as poking, and for proprioception. For proprioception, only the muscle spindle. So muscle spindle has got two types of sensory fibers, 1A and 2. Golgi tendon organ has got one type of sensory fiber, and that is 1B. Right? So A gamma is motor to the muscle spindle. Muscle spindle also has a motor supply, and that is A gamma. A delta is fast pain. It is meant for temperature. It is meant for some mechanoreception. This is type 3. B is preganglionic autonomic, does not have any number. C is postganglionic sympathetic, will not have any number. But C fibers can also be sensory. Sensory means for crude touch, itch, slow pain, temperature, cold and warm, pressure. This is your type 4. Type 4, right? Now, please remember, when you look at these things, now let's have a look at what is the susceptibility. Look at the susceptibility. When I say susceptibility, that means susceptibility of nerve fibers. Susceptibility of nerve fibers to, number 1, pressure. Susceptibility of nerve fibers to pressure, to hypoxia, and number 3, to local anesthetics. To local anesthetics, right? Let's see. Susceptibility of the nerve fiber to pressure, to hypoxia, and to local anesthetics, right? Now, Manendra Prasad, numerical classification is only meant for sensory fibers. Will autonomic fibers have a number? No, I've been repeating this. Motor fibers do not have a number. Autonomic fibers do not have a number. Postganglionic parasympathetic. Now, just remember your basics. Parasympathetic, postganglionic parasympathetic nerve fibers are extremely short. They do not exist as separate nerve fibers, because where are the parasympathetic ganglia located? In or near the organs they innervate. You cannot see them as a separate nerve fiber. So that is why it is not included. If you can see them, they would still be seen. Have you understood? The postganglionic sympathetic nerve fibers are long fibers. Postganglionic parasympathetic are extremely small, because the parasympathetic ganglia are located in or near the organ they innervate. Okay? All right. So susceptibility of the nerve fibers to pressure, to hypoxia, and to local anesthetics. Now, what does this mean? This means that if I apply external pressure, transmission will be affected in which fiber first? I'm not asking you which fiber is carrying the pressure. That's a separate question altogether. We are talking about susceptibility. Susceptibility means that if I apply external pressure, then the transmission will be affected in which fiber first, right? So if for pressure, the most susceptible fiber is A alpha, then A, then B, and then C. And out of A, it is the A alpha which is most susceptible. Right? A alpha becomes the most susceptible to external pressure. And let's see the clinical importance of knowing this. Clinical importance of knowing this is, see, more the diameter, more the susceptibility to external pressure. What is the, for example, Saturday night palsy, isn't it? Now, what happens on a Saturday night? Saturday night is usually a night of partying, right? That is, generally people party on Saturday nights, they drink a lot, they come home drunk, sleep with the arm on the chair, there is compression of the brachial plexus. Which is the fiber which is most susceptible to pressure? A alpha. And what is A alpha? Motor. So motor neurons are affected, and he gets up with the palsy of the affected side on the next morning. right? This is also known as the Sunday morning palsy. Why Sunday morning palsy? Because he realizes on Sunday morning what he's done, right? So it is the A alpha motor neuron which is the most susceptible to pressure, and A alpha is your somatic motor. So somatic motor. So A alpha most susceptible to pressure. Let's see what is the fibers which are susceptible to hypoxia. B fibers are most susceptible to hypoxia, then to A, and then C. To local anesthetics. Local anesthetics. Local anesthetics. Local anesthetics, again, A more than B more than C, right? A more than B more than C. But please remember, out of the A fibers, it is the A gamma which is most susceptible, right? A gamma which is most susceptible, not A alpha, but it is the A gamma which is most susceptible. C is in fact the least susceptible to local anesthetics. Please understand, least susceptibility does not mean that local anesthetics do not affect the type C fibers. That does not mean this. In fact, type C are your pain-carrying fibers. So anesthetics have to affect type C. But when I say most susceptible and least susceptible, what does this mean? This means that to block transmission in the type A gamma fiber, I need a lower concentration of the local anesthetic. To block transmission in type C, I need a slightly higher concentration of the anesthetic. That is all that it means. Right? So this is please understand, Shruti, there is no reason. This has been determined on an experimental basis. A gamma is the most susceptible to local anesthetics, and C in fact is the least susceptible to local anesthetics, right? Okay. Now, this is the susceptibility of the nerve fibers. By the way, least susceptibility to local anesthetic, this was asked as an AIMS question in 2018, right? This was an AIMS question in 2018. Right? Least susceptible to local anesthetics is type C. Now, 2-3 important points before we take on. So if you can see this, C fibers are the least susceptible to pressure. They are the least susceptible to hypoxia. They are also the least susceptible to local anesthetics. Have you understood? So they're least susceptible to all three, right? Now, 2-3 important points, and that is just factual points. There's nothing to understand here. How much are the total number of neurons? There are 100 billion neurons. 100 billion neurons. Just 2 or 3 facts here, factual points. 100 billion neurons, which are more the glial cells. Glial cells are much more than the total number of neurons. Glial cells are 3 to 5 times the total number of neurons. Yes? Another small little fact, and that is 40% of the total genetic material. 40% of the total genes are meant for the central nervous system, right? They are meant for the nervous system. The total 40% of the total genes is meant for the nervous system. So these are some simple points, factual points. Which are the most numerous? Which are the most numerous? Which are the most numerous? Is it going to be type A, type B, or type C? It is in fact the type C fibers which are the most numerous. So total number of neurons, 100 billion. Glial cells, 3 to 5 times the total number of neurons, much more than the number of neurons. 40% of the total genetic material is meant for the nervous system. Which neurons are the most numerous? Will be C. Okay? All right. Yes, Manu, thankfully CNS dominance. Thankfully, this keeps us straight, keeps us aligned. Yes, okay. Okay. All right. Now, all this is when we've done these new nerve fiber classifications, I'm going to do some simple questions with you just to reinforce what you have done in the nerve fiber classification, right? Now, First important point, which is a nerve, which is the fiber, which is a nerve fiber for fine touch, which is a nerve fiber for fine touch. Nerve fiber for fine touch is A beta, right? Nerve fiber for fine touch, A beta, which is a nerve fiber for crude touch. For crude touch is C. Fine touch and crude touch. Somebody was asking what is the difference between fine touch. Fine touch is, for example, if you take a cotton wool, you remember sensory system examination. In a first year, you were taught for fine touch, you use a cotton wool and that is fine touch. Crude touch would be just touching with the finger or with a pen or something. That is crude touch. So nerve fiber, which is meant for fine touch, is A beta. For crude touch is C. Which is the nerve fiber for pressure. Nerve fiber for pressure. The nerve fiber which carries pressure is C. Which is a nerve fiber which carries deep pressure. Nerve fiber which carries deep pressure, this is A beta. Deep pressure is poking. C is for pressure. Next question, which is a fiber for vibration? For vibration, the nerve fiber is A beta. Vibration, nerve fiber is A beta. Which is a fiber for itch. Itch is type C. Which is a nerve fiber for fast pain. For fast pain is A delta. Which is a nerve fiber for slow pain. For slow pain is C. Right? Slow pain is C. Next question. Which is a fiber for cold sensation? Cold sensation is A delta. Which is a nerve fiber for warm sensation? Warm sensation is A delta and C. Cold is A delta and warm is A delta and C. Let's see this. Which is the sensory or afferent fiber? Sensory or the afferent? Afferent from the muscle spindle. Afferent from the muscle spindle. Right? Sensory or the afferent from the muscle spindle. Is it going to be 1A? Is it 1B? Is it 1A and 2? Or is it 1B and 2? Now, sensory from the muscle spindle is 1A and 2. 1A, if you remember, was the same thing as A alpha. 2 is the same thing as A beta. Have you understood? These are synonyms. Yes? So 1A and 2. Sorry, A delta and C. Warm is only C. Just correct that, please. Cold is A delta and C. Warm is only C. Just correct that. Okay, thank you for bringing it to my notice. Okay, opposite. ठीक है. All right, sensory or afferent from the muscle spindle, 1A and 2. What is the sensory or the afferent from the Golgi tendon organ? Sensory or the afferent from the Golgi tendon organ? This is only 1B, and 1B was A beta. Have you understood? Synonym for 1B is the A beta, right? Okay. Then let's see another question here. What is motor to extrafusal muscle fibers? What is motor to the extrafusal muscle fibers? Motor to the extrafusal muscle fibers. And what is motor to the intrafusal muscle fibers? Intrafusal muscle fibers, right? Motor to the extrafusal muscle fibers. Motor to the extra, let's just see this. I've made a couple of mistakes here. Let me just clarify them once again. Cold sensation by A delta and C, warm sensation only by C. Sensory from the muscle spindle is 1A and 2. 1A is A alpha, 2 is A beta. Sensory from the Golgi tendon organ is 1B or A alpha, right? Motor to the extrafusal muscle fibers, this is A alpha. Motor to the intrafusal muscle fibers is A gamma, right? Motor to the extrafusal muscle fibers is A alpha, and motor to the intrafusal muscle fibers is A gamma, right? Now, what is extrafusal and what is intrafusal? Fusal means spindle. So extrafusal muscle fibers are outside the muscle spindle. These are your somatic motor, and intrafusal muscle fibers means inside the muscle spindle, and that is known as A gamma. Right? So let's see this once again. Let's see all these questions which I've done with you. Nerve fiber for fine touch, A beta. Crude touch, C. Pressure, C. Deep pressure, which is known as poking, A beta. Vibration is A beta. Itch is C. Fast pain is A delta. Slow pain is C. Cold is A delta and C. Warm is C. Sensory from the muscle spindle, 1A and 2. 1A is synonymous with A alpha, 2 is synonymous with A beta. Sensory from the Golgi tendon organ is 1B, which is the same thing as A alpha. Motor to the extrafusal muscle fibers, A alpha. Motor to the intrafusal muscle fibers is A beta. Tanvi, please see this. 1A is sensory from muscle spindle. Muscle spindle, not from Golgi tendon organ, is 1B. Yes, Megha, A alpha is the same thing as type 1, which is divided into 1A and 1B. 1A is from the muscle spindle and 1B is from the Golgi tendon organ. Okay? Okay. All right. So next is, so once we've done the classification of nerve fibers, I want to discuss with you in detail what do you understand by extrafusal muscle fibers and intrafusal muscle fibers, right? Extrafusal and intrafusal muscle fibers. Okay. Now, as far as extrafusal and intrafusal muscle fibers is concerned, we need to do what is the structure of the muscle spindle. Let's see the structure of the muscle spindle. Now, as far as the muscle spindle is concerned, muscle spindle is muscle spindle. Now, this is a receptor for muscle length, right? What is the muscle spindle? This is a receptor for muscle length, right? This is a receptor for muscle length. Muscle spindle is a receptor for muscle length. So let's have a look at the organization of the muscle spindle. Now, if you look at a muscle, if this is a tendon, this is the belly of the muscle, and this is another tendon. This is what a muscle looks like, right? This is the Kavya, A alpha is type 1. Type 1 is 1A and 1B. So I can call 1A A alpha. I can also call 1B A alpha. What is the confusion in that? Why are you again and again putting that same question? Isn't it 1A or 1B are both A alpha? What is the problem? Muscle fibers for crude touch, क्या मतलब हुआ बेटा? Crude touch तो sensation है. Sensation के लिए sensory fiber है. Sensory fiber for crude touch is type C or type. All right. Let's go on to the muscle spindle. Now, muscle spindle. Okay. Now, please see the muscle spindle now. Now, muscle spindle is a receptor for muscle length. So, if I look at a muscle, the muscle has got fibers which extend from tendon to tendon, right? These are fibers which extend from tendon to tendon. Muscle fibers have got, a muscle has got fibers which extend from tendon to tendon. And these fibers which extend from tendon to tendon, these are called the extrafusal muscle fibers. These are known as the extrafusal muscle fibers. extrafusal muscle fibers, and these fibers are responsible for muscle contraction, right? The muscle contraction. Extrafusal muscle fibers are responsible for muscle contraction. And what is the nerve supply to the extrafusal muscle fibers? The motor supply to these extrafusal muscle fibers is A alpha. We've discussed this. A alpha is the somatic motor. This is the spinal motor neuron. This supplies the extrafusal muscle fibers, which are responsible for muscle contraction. Right? Now, present within the muscle spindle, there is a receptor which extends from tendon to tendon, and this receptor, this is a spindle-shaped receptor. This is a spindle-shaped receptor, and this receptor is known as a muscle spindle. This muscle spindle also extends from tendon to tendon. I'm just showing you one muscle spindle, but of course there are numerous muscle spindles. Muscle spindle, like I said, is a receptor for muscle length. Right? This extends from tendon to tendon. So if there is an increase in the length of the muscle, there will be increase in the length of the muscle spindle. Right? Spindle extends from tendon to tendon. Right? Now, if I look at the muscle spindle, if I enlarge the muscle spindle and see, this is what it looks like. I'm enlarging just the muscle spindle now. Now, the muscle spindle has got an outer connective tissue sheath. Right? Outer connective tissue sheath. Outer connective tissue sheath. Yes. Now, inside this connective tissue sheath, you have the intrafusal muscle fibers. Right? You have the intrafusal muscle fibers. Inside fusal spindle. You have these intrafusal muscle fibers. Right? These are the intrafusal muscle fibers. Intrafusal muscle fibers. And which are these intrafusal muscle fibers? There are two types of these intrafusal muscle fibers. You have what are known as the nuclear bag fibers. There are the nuclear bag fibers, and you have the nuclear chain fibers. What is nuclear bag? Nuclear bag fibers have got nuclei in a dilatation in the center. So it looks like a bag. So there are nuclear bag fibers, and you've got nuclear chain fibers. Nuclear chain fibers where the nuclei are in a row. It looks like a new chain of nuclei. So it's nuclear chain fibers, nuclear bag fibers and nuclear chain fibers. Nuclear bag fibers are further subdivided into nuclear bag dynamic, nuclear bag dynamic and nuclear bag static. Nuclear bag dynamic and nuclear bag static. Now, what is dynamic and what is static? Dynamic means they detect rate of change. They detect rate of change of muscle length. Right? They detect rate of change of muscle length. Or you can also say they detect the velocity of change of muscle length. Right? Velocity of change of muscle length. Right? This is what is meant by rate of change. Please remember, whenever I use the term rate, it means it has to be per unit time. The length of the muscle is changing with time. That is the rate at which this change occurs. That is detected by the nuclear bag dynamic. For example, when I'm doing an extension flexion like this, there is a change in the length of the biceps, there's a change in the length of the triceps. Rate of change. Is it a fast rate of change? Is it a slow rate of change? That is detected by the nuclear bag dynamic. Whereas nuclear bag static, this detects. Nuclear bag static will detect a steady length. This detects a steady length. Nuclear bag static will detect a steady, this detects a steady length. Now, nuclear chain is only of one type, and nuclear chain is also meant, nuclear chain is for only the static response. Static response. So both nuclear bag static and nuclear chain static, they detect steady length. Steady length means static. For example, after I've finished doing my flexion extension, whatever is the length of the muscle when it is steady, static, that is my nuclear bag static and nuclear chain. So for dynamic response, it is the nuclear bag dynamic. For a static response, it is going to be nuclear bag static. For dynamic response, so if you're asked a question, what is for dynamic response? For dynamic response is going to be the nuclear bag dynamic. Nuclear bag dynamic. And for static response, that means to detect the length when the muscle length is steady, that is going to be nuclear bag static. Nuclear bag static and the nuclear chain. Nuclear bag static and nuclear chain. So this is for dynamic response, nuclear bag dynamic. For static response, nuclear bag static and nuclear chain. Now let us see the nerve supply. Now nerve supply for the nuclear bag dynamic, nuclear bag static and nuclear chain. Let's try and see that. This is nuclear bag dynamic. This is nuclear bag static. And this is the nuclear chain. Right? Let's see what are the sensory fibers, what are the motor fibers to these different nuclear intrafusal muscle fibers, nuclear bag dynamic, nuclear bag static, and nuclear chain. Now the sensory fiber from nuclear bag dynamic, this is the sensory fiber. This is 1A, and 1A is also known as the annulospiral ending. Annulospiral ending. Right? This is going to carry impulses from the muscle spindle to the spinal cord. This is a sensory fiber. 1A is a sensory fiber. This is what is known as the annulospiral ending. This is a sensory fiber which will carry impulses from the nuclear bag to the spinal cord. Nuclear bag static. Nuclear bag static is type 1A and 2. From nuclear bag static is 1A and 2. And what is 2? 2 is also known as a flower spray ending. Type 2 is also known as a flower spray ending. It looks like a spray of flowers. So for dynamic response, only 1A. For static, nuclear bag static, it is 1A and 2. From nuclear chain, it is again 1A and 2. 1A and 2. So for dynamic response, 1A. For static response, 1A and 2. What is 1A? Annulospiral. 2 is also known as the flower spray ending. Now, if you remember, these are also known as, these are muscle fibers. They are intrafusal muscle fibers. So that means they also have actin myosin. That means these fibers are also contractile, though they are not responsible for muscle contraction. But remember, they have actin and myosin. And where is this actin myosin present? Where are the light and dark bands present? They are present towards the ends of the intrafusal muscle fibers. The ends of these intrafusal muscle fibers have got dark and light bands. They have got actin and myosin. Please remember, actin and myosin are not present throughout the muscle fiber. They are present only towards the end. So obviously they will also, if they've got actin myosin, if they've got light and dark bands, they will also have a motor supply. And let's see what is the motor supply here. Motor supply is, now these are motor fibers. So if you can appreciate the direction of the arrow, motor fibers are carrying impulses from the spinal cord to the ends of the intrafusal muscle fibers. And these are known as A gamma. To the nuclear bag dynamic, it is going to be A gamma dynamic. To the nuclear bag static, it is going to be A gamma static. Right? A gamma static. And to the nuclear chain, it is again going to be A gamma static. A gamma static, right? So this is as far as the nerve fibers are concerned. From the dynamic, nuclear bag dynamic, 1A for nuclear bag static, 1A and 2 for nuclear chain, 1A and 2. These are the sensory fibers. Motor fibers will go towards the ends of these intrafusal muscle fibers. To nuclear bag dynamic, it is A gamma dynamic. To nuclear bag static is A gamma static. And to nuclear chain is also A gamma static. Now, that means if I ask you this question for dynamic response, we said nuclear bag dynamic, the sensory fiber is only 1A. For static response is 1A and 2, isn't it? For nuclear bag dynamic, for dynamic response, nuclear bag dynamic. For static response, nuclear bag static and nuclear chain. Sensory fibers for dynamic response, 1A. Sensory fibers for static response, 1A and 2. Now, the next important point is, what is the significance of these A gamma fibers? Okay, let's try and understand this. Now, the muscle spindle can be stimulated. Muscle spindle can be stimulated in 2 ways, in the following 2 ways, right? Muscle spindle can be stimulated in the following 2 ways. Remember, muscle spindle is a receptor for muscle length. Right? So how can a muscle spindle be stimulated? Muscle spindle can be stimulated in the following 2 ways. Number 1, if I increase the muscle length. If I increase the muscle length, for example, during a stretch reflex, we'll be doing stretch reflex in detail in the central nervous system. So when I increase the muscle length, I will also increase the length of the muscle spindle, because remember, the spindle extends from tendon to tendon. So if I increase this muscle length, if I stretch the muscle, I will be increasing the length of the muscle spindle and stimulating the muscle spindle. And second reason is if I increase the A gamma motor neuron discharge. If I increase the A gamma motor neuron discharge, remember, A gamma is supplying the ends of the muscle spindle. So if I increase the A gamma motor neuron discharge, this causes contraction of the ends of, this causes contraction of the ends of the intrafusal muscle fibers. It causes contraction of the ends of the intrafusal muscle fibers. Intrafusal muscle fibers, nuclear bag, nuclear chain, these will contract. The ends will contract. And the moment the ends on both sides will contract, this now stretches the receptor, the central receptor portion. This now stretches the central receptor portion of the intrafusal muscle fibers. It stretches the central receptor portion of the intrafusal muscle fibers. Right? It stretches the central receptor portion of the intrafusal muscle fibers, and that in turn stimulates the muscle spindle. Right? It stimulates the muscle spindle. Right? And what happens when the muscle spindle gets stimulated? We'll do it a little later in central nervous system. So, right? So muscle spindle can be stimulated in 2 ways. Number 1, if I increase the muscle length or stretch the muscle, for example, during a stretch reflex, or if I increase the A gamma motor neuron discharge. If I increase the A gamma discharge, it causes contraction of the ends of the muscle spindle. And if the ends will contract, the central receptor portion gets stretched, and that in turn will stimulate the muscle spindle. Right? All right. Okay. So see this once again. Nerve supply, the sensory supply from the nuclear bag dynamic, 1A and 2. Motor supply to nuclear bag dynamic is A gamma dynamic. For nuclear bag static, sensory nerve supply, for nuclear bag dynamic, only 1A. For nuclear bag static, 1A and 2. For nuclear chain, 1A and 2. Motor supply to nuclear bag dynamic, A gamma dynamic. To the nuclear bag static, it is A gamma static, and nuclear chain is also A gamma static. right? And what is the significance? Please remember, A gamma supplies only the ends of the intrafusal muscle fibers, not the entire intrafusal muscle fiber. Why the ends? Because the actin myosin are present at the ends. What is the significance? When the A gamma discharge increases, the ends of the muscle spindle will contract, and that in turn will stretch the receptor portion, the central receptor portion, and stimulate the muscle spindle. All right. This is as far as your the muscle spindle is concerned. Next, let's have further to this. This is just the basic that we're doing over here. What is the clinical significance? What causes increase in A gamma discharge? What happens when the muscle spindle is stimulated? We will do all that in central nervous system, right? Motor supply of the extrafusal muscle fibers has already been done. Upshare, that has been done so many times. That is A alpha. A alpha. Motor to the intrafusal muscle fibers is A gamma, to extrafusal muscle fibers is A alpha. What is the significance? I told you I will do it in central nervous system. Yes, what is A motor neuron discharge? We'll be doing it in central nervous system. There are special tracts which are called reticulospinal tract. Proprioception is proprioception, Jaideep. It means position sense, right? Whether it is carried in type 1 or in type 2 fibers. It doesn't matter. It is the speed which will be different. That's all. Okay. Now, the next important point, the next thing that we're going to take up is what happens if there is okay. Now, next, let's see, what do you understand by the Wallerian degeneration and regeneration? Let's see this. Wallerian degeneration. Wallerian degeneration, right? This was first described by Waller, yes. It's first described by Waller. And what happens in a Wallerian degeneration? Now, firstly, you must know what is the Seddon's classification of nerve injuries. Seddon's classification of nerve injuries. Seddon's classification of nerve injuries. Right? Now, what is Seddon's classification? None. You have neuropraxia. Neuropraxia, please remember, is only a functional disturbance, right? Neuropraxia, exontemesis, and neurontemesis. Neurontemesis, yeah? Exontemesis and neurontemesis. Now, this is your neuropraxia. This is known as Seddon's classification of nerve fibers, right? This is going to be neuropraxia, neurontemesis, exontemesis, neurontemesis. Now, neuropraxia is a functional disturbance. This is commonly your pins and needles, which you are sitting in one place for a long time, for example, during a class, and you get that pins and needles sensation. That is called neuropraxia. This is just a functional disturbance, right? There is no degeneration that you're going to see over here. This is only a functional disturbance. So no degeneration. Is there going to be any degeneration in the case of neuropraxia? No. Exontemesis. Here the neurilemma is intact. Neurilemma is intact. And what is neurilemma? Neurilemma is the outermost layer of the Schwann cell. Will there be a degeneration seen here? Yes. Will there be a regeneration seen here? Yes. Right? In neuropraxia, there is no degeneration, there is no regeneration. But in exontemesis, there is a degeneration and there is a regeneration. In neurontemesis, the neurilemma itself is destroyed. The neurilemma itself is destroyed. So there is a degeneration, but there is going to be no regeneration. So please remember, for regeneration to take place, neurilemma should be intact. What is neurilemma? Neurilemma is the outermost layer of the Schwann cell. Outermost layer of the Schwann cell, right? Now, let us see what are the changes, what will happen when in Wallerian degeneration, and how does the regeneration take place. Yes. Now, when I look at so please remember, for regeneration to take place, regeneration occurs if, number one, neurilemma is intact. Yes. Neurilemma is intact. Neurilemma must be present for regeneration to take place. And number two, the distance between the cut ends, has to be less than 3 millimeters apart. Then the regeneration can take place. Otherwise, we will not see any sort of regeneration. Please remember, neurontomesis, exontomesis is not just seen in injuries. You also see in conditions which affect nerve fibers. For example, diabetes, diabetic neuropathy, autonomic neuropathy, right? These will start affecting the nerve fibers. So let's see, regeneration will occur if the neurilemma is intact and difference is distance between the cut ends is less than 3 millimeters. So let us see what are the changes which are going to take place. Now, if you look at a nerve fiber, right? This is a myelinated neuron here. Let's say there is a cut injury. Yes. Let's say there is a cut injury over here. Yes. Now, how do we describe the changes which are going to happen? What is going to happen in a degeneration? Let's first see what are the changes in the cell body. Changes in the cell body. What are the changes in the cell body? Within the first 24 to 48 hours, changes in the cell body, in 24 to 48 hours, in the first 28 to 48 hours, you get what is known as chromatolysis. There is chromatolysis. There is a disappearance. There is a disappearance of the Nissel's granules. Disappearance of the Nissel's granules. What are Nissel's granules? Nissel's granules are nothing but ribosomes and the mRNA. Right? So there is disappearance of the Nissel's granules, which is known as chromatolysis. This is the changes which occur in the cell body. There are these Nissel's granules which are present. These are ribosomes. These disappear. So there is chromatolysis. Right? Then also the second change which will occur is the nucleus moves to the periphery. Nucleus moves to the periphery. Nucleus moves to the periphery, right? So within the first 24 to 48 hours, you have what is known as disappearance of the Nissel's granules, and nucleus will move to the periphery. Then let us see what are going to be the changes in the proximal changes in the distal segment. What are the changes in the distal segment? Which is the distal segment? Distal segment means which is distal from the cell body. That means this segment. This is the distal segment. What are the changes in the distal segment? Within a few hours of injury, there is a swelling of the axis cylinder. Swelling of the axis cylinder. Axis cylinder means the axon per se. There is a swelling of the axis cylinder. In 3 to 5 days, you get an axonal degeneration. The axon begins to degenerate. There is an axonal degeneration which is going to be seen. Within 3 to 5 days, first week itself, within a few hours of injury, the swelling starts, followed by degeneration, the axonal degeneration. The 8th day, after the first week is complete, the myelin degeneration starts. myelin degeneration starts, right? And by the 32nd day, myelin degeneration is complete. Myelin degeneration is complete. Right? Myelin. So these are the changes which are going to happen. Within a few hours, there is a swelling of the axis cylinder followed by degeneration. Then the 8th day, the myelin degeneration will start. And on the 32nd day, myelin degeneration is going to be complete. Right? And now macrophages, they remove cellular debris. Macrophages will remove the debris, and you get an empty tube of Neuri lemma. There is an empty tube of Neuri lemma. Empty tube of Neuri lemma, which is known as a ghost tube. Empty tube of Neuri lemma is formed, which is known as a ghost tube. Right? So please write this down, right? Vismandeep Kaur, Nissel's granules are ribosomes. Ribosomes are associated with mRNA, messenger RNA. Significant to know is, Deepu, you are asked questions in your exams. Okay? All right. Now, degeneration, yes, Sakshi Potidar, degeneration is, we are talking about degeneration because we also have to talk about regeneration. Degeneration can occur in all types of neurons, but regeneration occurs only in myelinated neurons and only if the Neuri lemma is intact. Neuri lemma is the outermost layer of the Schwann cell. Schwann cell will be associated only with myelinated neurons. So this is formation of a ghost tube. Right? Macrophages will remove the debris and there is an empty tube of Neuri lemma which is formed, and that is known as the ghost tube. Now, what are the changes in the proximal segment? Changes in proximal segment. Changes in the proximal segment are similar to changes in the distal segment. They are similar to distal segment, but up to, similar to the distal segment, but up to the nearest node of Ranvier, up to the nearest node of Ranvier. Nearest node of Ranvier. Nearest node of Ranvier means nearest to the site of injury. These are going to be similar to the distal segment, but up to the nearest node of Ranvier. Now, let's see this over here. I said, what are the changes in the cell body? Within the first 24 to 48 hours, chromatolysis. Nucleus will move to the periphery. Then what are the changes in the distal segment? Within a few hours, swelling of the axis cylinder. Axis cylinder means the axon, right? That begins to swell up, and by the 3rd to the 5th day, axonal degeneration will start. On the 8th day, the myelin degeneration starts, and 3 to 5 days, axonal degeneration occurs. 8th day, myelin degeneration starts, and myelin degeneration is complete by the 32nd day, right? So in other words, just to remember this, first day itself, within few days of first day itself, swelling. First week, axonal degeneration. After first week, myelin degeneration will start. After 1 month, myelin degeneration is going to be complete, right? So after 1 week, the myelin degeneration will start. After 1 month, myelin degeneration is complete. Within the first day itself, swelling. First week, 3 to 5 days, axonal degeneration. After first week, myelin degeneration starts, complete by the first month, right? So if you can see this over here, now I said changes in the proximal segment up to the nearest node of Ranvier. Nearest node of Ranvier to the site of injury is this one. So in the proximal segment, the changes will be seen over here, up to the nearest node of Ranvier. Okay? Now, if I draw this for you, if we see this sequentially, let's see. Now, this becomes important because you can be asked a question that arrange them sequentially. You know that in AIM especially, they ask you sequential arrangement. So this is one of them. Ultimately, it will result in what is known as a ghost tube. right? So if you can see this here, right? This is a myelinated neuron. Let's see, there is a cut injury. There is a cut injury, let's say over here. Here you have the Nissel's granules, yes? So the first change which will occur in the cell body, disappearance of Nissel's granules. No Nissel's granules are going to be seen. Nucleus will move to the periphery, yes? This is what happens. This is what is meant by a ghost tube. It is an empty tube of neurilemma, which is called a ghost tube. This is what is going to happen, right? This is the same duration of time, Jahanavi? Yes, okay. Yes, CP. What do you want me to see in the chat box? I can't scroll up a little. What do you want me to do? What do you want me to? What do you want me to tell you? Neurilemma is the outermost layer of the Schwann cell. Neurilemma is the outermost layer of the Schwann cell, which is a living part of the Schwann cell. Yes, both the proximal and the distal parts, the changes will occur simultaneously. All right. Okay. So this is as far as your formation of a ghost tube is concerned. Now, regeneration. Let's have a look at the regeneration. I've already told you when will the regeneration occur. Regeneration will occur if the outermost layer, neurilemma, is intact. Now let us see the regeneration part. Yes, outermost layer of the Schwann cell should be intact. Neurilemma should be intact, right? And if the distance between the cut ends is less than, this is the distance between the cut ends, this should be less than 3 millimeters. Yes, then you get a regeneration. Distance between the cut ends should be less than 3 millimeters. Then regeneration will occur. How does the regeneration occur? Now, the first thing which happens is there is the axonal stump. Axonal stump. Axonal stump, there is a sprouting of the axonal stump which is seen. There is a sprouting of the axonal stump which is seen. Sprouting of the axonal stump which is seen. This now enters into the ghost tube. It enters into the ghost tube. So the connection is re-established. Myelin is laid down. Second thing which happens is myelin is laid down in sheets. Myelin is laid down in sheets. Not that typical arrangement where the Schwann cell went wrapped around the axon. That will not happen. Myelin is laid down in sheets by the neurilemma. Right? So sprouting of the axonal stump, when I say sprouting means it sort of sprouts like this, it enters into the ghost tube and there is a connection which is re-established. It grows into the ghost tube and connection is re-established. Myelin is laid down in sheets. Yes? Now, what is the rate of regeneration? The rate of regeneration, what is the rate of regeneration? This is going to be 1 millimeter per day. 1 millimeter per day or 1 inch per month. This is the rate of regeneration, 1 millimeter per day or 1 inch per month. Regeneration will be complete in. Regeneration is complete, is usually complete by 1 year. If there is no regeneration by 1 year, then we can take it for granted that regeneration is not going to happen. So it's usually complete by 1 year, right? This is the rate of regeneration. Are we clear with that, right? So this is as far as the degeneration and regeneration is concerned. Once again, now, important question which is asked from here. You are asked a question, what is the first change in Wallerian degeneration? What is the first change which occurs, right? Question says, in Wallerian degeneration, which is the first change which is going to be seen? Which is the first change which is seen? Is it going to be chromatolysis? Is it going to be chromatolysis, or is it going to be the axonal degeneration? Right? Axonal degeneration. Chromatolysis or axonal degeneration. Is it going to be A or is it going to be B? Now, I've already told you chromatolysis is 24 to 48 hours. Now, axonal degeneration starts within a few hours, right? It starts within a few hours of injury. It starts within a few hours. How does it start? With a swelling, right? But it takes 3 to 5 days for the degeneration to happen. It takes 3 to 5 days. So if you are asked a question, what is the first change? My better answer, now swelling is not there. Swelling of the axis cylinder is not there in the options. It says axonal degeneration. In that case, my better answer becomes chromatolysis, isn't it? Axonal degeneration is taking 3 to 5 days, whereas chromatolysis is occurring within the first 2 days. It's complete within the first 2 days. Yes, starting of axonal degeneration, yes, within a few hours, but it does not say starting. It just says axonal degeneration. So that means an axonal degeneration takes 3 to 5 days to complete. So a better answer here becomes chromatolysis, right? Okay. Hafsa Yunus, regeneration is only if the distance between the cut ends is less than 3 millimeters. But the whole nerve has to regenerate now. Whole nerve has to regenerate, which will take time, 1 millimeter per day. There's a difference between the two things. Distance, see what happens when there are nerve injuries. What is the first thing the surgeon does? He tries to bring the cut ends as close to each other as possible so that regeneration can happen. Have you understood? Right? Rate of regeneration is one. Nucleus will go back into its position. Nucleus will go back to its position. Nissls granules will reappear. Okay. Yes. Yashaswini, in a heart is supplied by autonomic nerve fibers, isn't it? Autonomic nerve fibers. Post-ganglionic, sympathetic and parasympathetic, isn't it? So there is no question, those are unmyelinated, no regeneration. Okay, all right. So this is as far as your Wallerian degeneration and regeneration is concerned. Okay, yes, if there is a swelling of axon is given, then of course you can answer that because that occurs within a few hours. I've specified that, so there should not be a problem. Okay, all right. Let's see axoplasmic transport. The next is axoplasmic transport. Now, what is axoplasmic transport? Axoplasmic transport. Now, what is axoplasmic transport? Axoplasmic transport, this refers to the physical transport of substances. It refers to the physical transport of substances. This refers to the physical transport of substances through the axoplasm, right? It refers to the physical transport of substances through the axoplasm. That is known as axoplasmic transport. This is not the conduction of the action potential. Remember, conduction of action potential is electrical phenomena because of movement of charges, right? This is a physical transport of substances through the axoplasm. That is what is known as axoplasmic transport. physical transport of substances through the exoplasm. Remember, like I said, what is conduction of action potential? Conduction of action potential is electrotonic. That is not what you mean by exoplasmic transport. Conduction of the action potential is an electrical phenomena. It is due to movement of charges. This is electrotonic. Exoplasmic transport refers to the physical transport of substances through the exoplasm. Now, for exoplasmic transport, what is required? Exoplasmic transport requires number one, calcium. It requires the presence of calcium. It requires the presence of calcium. It requires the presence of ATP. It requires the presence of microtubules. It requires microtubules. Now, what are these microtubules? What are these microtubules? Microtubules, these are hollow tubes. These are hollow tubes. They are made of proteins. They are made of proteins called tubulins. There are two tubulins, that is alpha and beta, right? Microtubules, these are hollow tubes. They are made of proteins called tubulins, and tubulins are of two types. This is alpha and beta. And these alpha, beta, they alternate with each other. The alpha, beta, they will alternate with each other to form a ring. If this is alpha, beta, alpha, beta, alpha, beta, they will alternate with each other and form a ring. Yes, form a ring. And these rings, they are stacked one on top of the other. They are placed one on top of the other, and that forms a hollow tube, right? That forms a hollow tube. Are we clear with that? Right? These rings, they are stacked one on top of the other, and that forms a hollow tube. And that is known as a microtubule. So microtubule, these are hollow tubes. They are made of proteins called tubulins, that is alpha, beta. Alpha, beta alternate with each other and they form a ring, and these rings are placed one on top of the other, and that forms a hollow tube, which is known as, this is known as a microtubule. So a microtubule has got two ends. It has got two ends, and what are these two ends? And these two ends are the plus end. This is the plus end, or also known as the assembly end. Plus end or the assembly end, and the minus end, or what is known as the disassembly end. Plus end or the minus end. Plus end and the minus end. Plus end is known as the assembly end, and minus end is known as the disassembly end. What is assembly end? Assembly end means where the microtubules, where these rings are added, right? Remember, microtubules are dynamic structures. When you say dynamic, that means they can be modified. Yes, there is addition of these rings, removal of these rings. Plus end is the end where the rings are added, and minus end is the end where the rings are removed. So they have a definite polarity. There is a plus and there is a minus end, right? So again, what are these microtubules? These are hollow tubes. They are made of proteins called tubulins. They have two ends, which is a plus end and a minus end. And these microtubules, they serve as tracks. They serve as tracks. And what are tracks? Literally, what is a track? Track means like a rail ka patri. Have you understood? Within the exoplasm, you've got these microtubules which are serving as tracks, and moving on these tracks are what are known as the molecular motors. Molecular motors. What are molecular motors? Molecular motors are small garis literally, which transport substances from one end of the neuron to the other. And what are these molecular motors? These molecular motors are kinesin, dynein, and myosin 5. Kinesin, dynein, and myosin 5. Right? Kinesin, dynein, and these are molecular motors which move on microtubules. Yes, move on microtubules. Microtubules are serving as tracks. Microtubules are present in the exoplasm. They are involved in exoplasmic transport. What moves in the microtubules are these motors. And what are these motors? Kinesin, dynein, and myosin 5. What do I need for exoplasmic transport? I need calcium, I need ATP, I need microtubules, I need molecular motors. Molecular motors. Now, what are the types of exoplasmic transport? Types of exoplasmic transport. What are the types of exoplasmic transport? There are two types of exoplasmic transport. There are two types of exoplasmic transport. There is an anterograde transport. There is an anterograde transport and there is a retrograde transport. Anterograde transport and retrograde transport. And what is anterograde and what is retrograde? What is anterograde and what is retrograde? Anterograde means it is away from the cell body. Retrograde means towards the cell body. Anterograde and retrograde. Yes, this is towards the cell body. Away from the cell body is anterograde and retrograde means towards the cell body. Now, anterograde transport is further subdivided into fast anterograde and they can also be divided into fast and slow anterograde. Right? Fast anterograde and slow anterograde. Retrograde transport is always fast. Fast anterograde, this is at a speed of 400 millimeters per day. I think the kind of messages that you're giving, I'm just getting a little disturbed by this. Please go ahead and take a break. Yes, I'll do this topic after the break. Let's take a break for 10 minutes. Yes, take a break for 10 minutes. Come back so that you are happy that you've got your break. You don't trouble me anymore, and we can all continue in peace. Okay? Take a break for 10 minutes. We will restart at 11:15. Okay? Okay.